After understanding about the basics of DNA fingerprinting, let us understand the process with the help of which this DNA fingerprinting is carried out. So let us understand about the making of DNA fingerprints. It is a laboratory procedure that helps in carrying out the DNA fingerprint. This process involves various sequential steps. Now, in order to start the process, it is necessary that we should collect the sample. Yes, so the first step begins with the extraction of the sample of DNA from either blood, semen, hair bulb, or any other such cell. Following it, the second step that comes is restriction digestion of DNA with the help of specialized enzymes, that is restriction endonuclease. Which is followed by the third step, that is after cutting or restriction digestion of DNA, it is necessary that these DNA fragments should be separated. Thus, separation of DNA fragments is carried out by process called electrophoresis. Depending on the type, the electrophoresis procedure is carried out. But mainly, agarose gel electrophoresis is used for the separation of such fragments. With the help of which the positive and negative pole attract the corresponding charge segments towards it. Following it, the DNA band that is obtained is transferred to a nylon membrane or nitrocellulose membrane. Further, the final step that comes is a radioactive DNA probe is formed. It is this DNA probe that binds to specific DNA sequences that are present on this nylon membrane. Thereafter, the DNA that remains unattached gets washed off, that is, gets separated off. This figure depicts the process of DNA fingerprinting as it can be seen within steps. The first step, extraction of the DNA sample. Second, which is followed by the cutting and separation of the DNA fragment using restriction endonucleases. Further, this DNA that is separated is transferred onto nitrocellulose or nylon membrane. Such a DNA pattern is observed on the nylon or nitrocellulose membrane. As a result of which, the radioactive DNA probe is obtained, which gives a specific band pattern of specific DNA sequences. Finally, this radioactive DNA probe is transferred on an X-ray film by direct exposure. As a result of which, the DNA band pattern that shows fingerprint can be viewed easily in the form of dark and light bands. That is by exhibiting a unique pattern of dark and light bands. This figure depicts the separated DNA fragment and its result from both paternal and maternal chromosome, which are compared from the respective chromosome number, and its fingerprint is carried out in the form of tandem repeats for the same chromosome number that was extracted from the maternity and paternity side. Now let us understand about the applications of the DNA fingerprinting. The first and the foremost application that is very important, we have already studied, that DNA fingerprinting plays a huge role in human and health society. That provides a justice system. By identifying the individuals who carries out crime, then various settling paternity issues, apart from that, it also helps in understanding the inheritary diseases and helps in monitoring the bone marrow transplant. It also helps in diagnosing various inheritary diseases like cystic fibrosis, then Alzheimer, sickle cell anemia, and many others. Thus, in this way, various inheritary disorders can be detected and cured with the help of DNA fingerprinting.